small comment to that, and that was really interesting. When the European Pharmacopoeia took up these NBC compounds, they said that normally for the full characterization of this product, the physicochemical is highly important, but not enough. And that is probably as well the value. On the one hand side, you need that, but you need a stepwise approach probably to go ahead with that. So the books are not close to that.
Well, Gert, thanks again. And last but definitely not least, we move to the other side of the ocean again. Sesha Neervannan from Allergan will talk about another set of complex drug products, ocular emulsions. Please take the floor.
Thank you, Don. I realize I'm between you and an exciting panel discussion. I'll try to keep it as quickly as possible so we can get on with a lot of questions. What I wanted to thank you, CLINAM, for the opportunity first, and what I wanted to elaborate today a little bit is a case study of what another one of the categories of complex drugs looks like, and the struggles we are in general having because there is no clear regulatory framework in approving these products. So in that aspect, ocular emulsions is one of the newer category of drugs, as I just saw in one of the slides. There was a product that a guidance was issued a few years ago. Not only is this, we think it's a complex product, but as you see in my title, it's also delivered to a very complex organ, which is the eye. And the disease is a very, very complex disease, dry eye, which is a confluence of many different mechanisms, immunomodulatory mechanisms in an eye with multiple tissues. So it's in totality, to me, it's a little bit more of a bigger category in terms of complexity. Not only is the product complex, the disease is complex, the organ that we're delivering to is complex. So that's one of the things that we are seeing as we are learning more and more about these products. I will not go into a definition again. I think you've heard the definition of what an NBCD is. But going to emulsions, emulsions are really complex dosage forms. They are typically in a dispersed state, and typically two immiscible liquids are mixed, and they are stabilized by a surface active agent, and the drug is then distributed. So typically it has multiple phases, not 10 to the 26 different phases, but still, enough of these phases where the drug can be distributed in all of these phases, and the drug can be in an oil phase or an aqueous phase and a micellar phase, so it can be in different phases, and the ratio of the drug in these phases are dynamic in nature, and it can change based on external environment, as well as it can be different as you have it in a vial that you're stored in your counter, and it can change as soon as you apply it into the target organ, especially the eye. As soon as it hits the eye, the drug distribution actually changes within these phases. And of course, one of the key attributes of a non-biologic complex drug is that the manufacturing process influences this aspect as well. Simple manufacturing steps can change drastically how the drug is distributed, and how the drug is distributed is critical to determine how the drug is available for efficacy and safety considerations. And that's one of the key aspects that we look at. So the product in question is Restasis. Restasis is a cyclosporin ophthalmic emulsion. It has immunomodulatory as well as anti-inflammatory effects through multiple different pathways. And it's been approved for increase in tear production in patients who have very poor, the tear is suppressed due to ocular inflammation. And it was a very challenging molecule to formulate. It was, you could not get into a standard ophthalmic formulations. Drug was not available. It was not efficacious. So it had to be formulated in an emulsion. And it was actually approved filed in 2001 and then approved in 2002 and launched in 2002. So about 3 years ago, FDA issued a draft guidance on a bioequivalence with a generic pathway, an ANDA pathway for this. And when we saw this, it was a very classic OGD issued pathway, a guidance pathway, where they said because so one of the key attributes for an ocular drug is that you cannot do bioequivalence studies. You cannot actually measure because your systemic bioavailability, the amount of drug in plasma is so small, you cannot really do a plasma pharmacokinetic study. You cannot sample tissues in the eye for obvious reasons. And so it's very hard to really do a pharmacokinetic bioequivalence. And so the FDA said you cannot do pharmacokinetic bioequivalence. Doing a dry eye clinical equivalence is really hard because it's a tough disease. And so because you cannot do it, an in vitro pathway is all you need. And all you need is a Q1, Q2 comparison, and they threw in a few physicochemical parameters just to say that, oh, we are throwing in some extra. And so this was issued, and when we looked at it, we said, no, this is a little bit more complex. You cannot take this particular pathway for a lot of reasons. And why is that? As I mentioned, it's already a complex delivery system. It treats a complex disease. It's local delivery. It's not systemically delivered. It's not systemically bioavailable. It's actually delivered locally to the tissues directly. And then the absorption pathway to these local tissues is very complex. It's not in linear drug release profile. There is a very short residence time. If you're familiar with ocular delivery, you put a drop in your eye, the amount of time the drug actually stays in the eye is less than 30 seconds. In fact, sometimes even less, and it's actually drained by the nasolacrimal drainage system. So the contact time is very, very short. And the other complexity is the drug, the disease state can actually significantly alter the absorption pathway, because one of the issues with dry eye is that not enough tear is produced. And if not enough tear is produced, the ocular surface parameters are very, very different. Just to give an idea, so you apply a drug in the precorneal area here. It can go into many tissues. It's not just a single pathway. It's not going into one singular compartment of the body. It actually distributes into multiple different pathways. And what we also have from a complexity standpoint is what's marked as E is what we think or where you need the drug for efficacy. So it's not just addressing going to a single tissue that you need efficacy. You actually need drug levels in multiple tissues to get the effect that you want from a clinical efficacy standpoint. And of course, there are other tissues you have to measure, including systemic circulation, for safety benefits. So as you can see here, it's a very widely distributed drug in terms of tissues, and you need to know exactly where you need the drug. The problem is there is no in vitro, there's no PK/PD that is understood for this drug nor for this disease. It's really a very tough question to answer, how much you need in each of these tissues for the safety and efficacy of the drug. And as well as the tear film actually has a huge impact on this particular formulation in terms of how it is released, how the drug is released. The salts in the tear is actually very critical in terms of breaking down the emulsion and releasing the drug. And of course, in a patient with dry eye, the tear film is not only less, but also the composition of the tear film is so different. The emulsion is not necessarily broken down the same way in every patient because their composition varies from patient to patient. So with all these complexities, we said, this is not, so we somehow have to convince the agency, we have to start a dialogue to convince them that this is not a simple generic pathway that you can adopt here. It has a big impact in terms of safety and efficacy if you don't characterize it properly. So rather than make theoretical arguments, we took an approach where we said, okay, let's just actually provide some data to the agency as part of providing feedback to the guidance document. And since we knew that the manufacturing process has a huge impact on the property of the drug, we decided to wear a hat of a typical generic company and said, okay, let's just make the product as the guidance states it. The guidance states it has to be qualitatively and quantitatively the same. So we said, okay, let's just keep that. Let's make the same formulations, but let's change the manufacturing processes. And when I say manufacturing processes, we change some very simple things like order of addition, how you add the different components during a manufacturing phase, how do you mix the shear rate, at what temperatures you mix. So we changed some simple parameters within the same manufacturing framework. We didn't drastically change the manufacturing process, made enough changes just within the framework and said, okay, let's just characterize these products. And so when we started characterizing these products, we took the guidance document as they have it and tested exactly as a generic sponsor would test their product. And when we looked at it, it was the product as we designed it was qualitatively and qualitatively similar, and the FDA defined a few of these parameters, physicochemical parameters, and when we measured these physicochemical parameters, so we had about 10 different formulations of restasis equivalent products, and in all of these properties, all of these formulations were they were essentially equivalent compared to restasis itself. So we satisfied every aspect of what the FDA guidance did. But then we said, let's take one step further and further our argument, our hypothesis that even though they meet these guidance documents, they're really not equivalent products. They're really not equivalent because it doesn't behave exactly the same way. So we decided to go a step further in terms of physicochemical analysis, looking at how sophisticated analytical techniques will measure the drug levels, as I mentioned, at different phases. Remember, that was not a criteria that FDA suggested in the guidance. And as well as we said, okay, let's assess the safety and efficacy of the product in an animal model. And that's the only thing we had. We could not do a kind of a study that we wanted to do in humans for obvious reasons. We can't do PK bioequivalence. But in an animal, we could actually show PK parameters in an animal study. And when we did this, we measured the drug distribution in multiple phases. We measured rheology profile, some extra additional things that we could do from a physicochemical standpoint. And we also looked at drug safety in animals and PK differences in certain tissues. And what did we see? So we saw that about, out of the 10 formulations, there were at least about 3 or 4 that had a different physicochemical profile. In the extended characterization and the sophisticated analysis we did in the drug distribution phases we did, clearly the manufacturing process changes impacted how the drug is distributing in these different tissues. And in addition, when we compare the pharmacokinetic parameters, you can see that there were clearly differences in terms of the pharmacokinetic parameters in rabbits and dogs, where some of these test formulations were between about the same as restasis to about 9-fold higher in terms of its exposure in certain components, in certain compartments of the eye, as well as in the area under the curve and Tmax. So all of these pharmacokinetic parameters, there were about 10-fold difference in terms of exposure, even though these formulations met exactly the same parameters that the FDA said you need to meet to show equivalence. In addition, when we did tolerability studies, some of these formulations that had high exposure in certain tissues, remember, these were multiple tissues. We measured tears, we measured cornea, we measured conjunctiva, we measured lacrimal glands, we measured a whole bunch of other tissues. And we found that certain levels, certain exposure beyond a certain point in certain tissues also caused significant tolerability. Some of these rabbits, they just shut their eye and they wouldn't open for about 2 hours. And that's usually an indication of severe irritation, severe discomfort in the animals. And so we showed that really this is not even though these formulations appear to be equivalent, they are really not equivalent. They really have an impact on effectiveness as well as safety of the product. And so that was clearly one of the things that we said, and we went back to the agency and said, look at this, your criteria is not sufficient. In vitro may not necessarily predict because we have gaps in knowledge. We don't have enough research in the area. There's not a whole lot of understanding of how these work, and so on and so forth. And the result of that was there was a lot of back and forth, and the agency acknowledged to some extent what we argued, and they reissued a guidance early this year. They still said you can still do in vitro characterization, you don't need in vivo characterization, but they went a few steps further, and I'm not going to read through this, but just to highlight, so you see my highlights in yellow, these were the changes that they proposed to what they had issued 3 years earlier. Essentially what they did was they said you have to do additional physicochemical characterization, you have to do more sophisticated analysis. The physicochemical attributes are not sufficient in what we had initially said. But one of the key things that they said is the last highlighted item here is that, and I can read, if you can't read this part, it says the methodology used for this in vitro drug release testing should be able to discriminate the effect of process variability in the products of test formulation. So essentially they acknowledge that manufacturing processes are very important. So we are still having these discussions. They recently just issued these guidances. They did acknowledge that this is still a first step in the process. And one of the things that we are still discussing is that, okay, this is fine, this is fantastic, we are making progress in our discussion, acknowledging that this is a complex product. However, there is still a link missing. So what do these parameters mean, the physicochemical parameters? How do they link to in vivo performance? Are you really measuring the right parameters? And then also, in some cases, some of the tests they had asked us to do, one of which is an in vitro drug release test. There is no in vitro drug release test in the literature for these types of products, especially in the eye. Remember, I said it stays on for 30 seconds. Most in vitro drug release tests come from an oral dosage form where you measure for at least about an hour or more. So clearly there are gaps in not only understanding of in vivo performance, but also gaps in vitro testing of these type of products. And so that's where we have, and then clearly understanding locally acting ophthalmic emulsions is important, as well as we suggested that there are research that is needed, and FDA actually just recently announced grants for some of these areas, these exact areas actually. They have proposed for whoever wants to do research, they have offered grants for these types of areas. And so the science is still evolving, and we are actually helping to build the science. But the fact is that there is no pathway. We are trying to use the existing pathway to fit these products into the current pathways, and clearly there's more dialogue needed, and clearly there's more discussions needed, and acknowledgement that you need to have a much more robust way of approving these drugs when it comes to patient safety elements. So with that, thank you very much. I know I exceeded my time a little bit, but thank you.
Any questions? Sesha, did you publish these results? I mean, you know my position there.
Yes. Yeah. So we have published parts of it. In fact, FDA published a paper arguing counter that some of these in vitro data is sufficient. We actually rebutted that position, and we presented some of that data. Of course, a lot of the in-depth data went into our NDA filing, which is not public yet. But we have been preparing to publish some of the actual data. It's not out there yet, but it is going to be in short order.
Okay, Marissa.
Very quick. It's a question, even though I know the response. Did you think of filing for disease model at the agency, at the European Medicine Agency qualification process?
Yes.
You know, we have a process specific,
yes,
and we are very strong in ophthalmic discussions.
Correct. And this product is not approved in Europe yet, and that's because there are still negotiations in terms of what would be a approvable pathway.
I'm not speaking about the product.
Okay.
I think you did a fantastic work with the various variations in the manufacturing process to explain that the manufacturing process has a direct impact on the performance of the drug. Now, my question is, did you consider submitting an in silico model based on the 10 different parameters you changed in order to have a model which can be used for emulsion in general?
No.
It's not product specific, it's a method qualification.
So no, we haven't, so we don't have, so obviously this product was approved more than 15 years ago. So we haven't had another clinical study to show the manufacturing differences yet. So these data were generated about a year and a half ago. So clearly when we develop another ocular emulsion, that would be, as you mentioned, as you develop a novel product, as you develop it, provide a pathway in terms of understanding the manufacturing processes, set some parameters so you can come back and say how this makes, how is this relevant from a clinical effect standpoint. But no, we haven't had a chance to go back and do that within this particular case yet. Yeah. But that's essentially what the approach is going to be moving forward, just as with the biologics, as you mentioned. Yeah.
Frank, please. Scott, Gerrit, please take a seat.